Hello guys, welcome to our channel AskPrep. Today's series is for 11th class biology. We will be studying chapter 11 transport in plants in detail. We have also added important notes for your exam preparation. All the best for your exam. Starting with what is translocation. Translocation refers to the long-distance transport of substances within plants, primarily through the vascular tissues, xylem and phloem. Unlike short-distance transport methods such as active transport, cytoplasmic streaming, and diffusion, translocation moves water, minerals, and organic compounds over greater distances to sustain plant growth and metabolism. Let's understand in detail what is translocation.
Transporting plants. Every cell in our body needs many different substances to function properly, for example, glucose and oxygen. But the cells cannot obtain the substance directly from outside. Therefore, they rely on the circulatory system to carry these substances from the body part that provides them, like lungs and small intestine, to all the body cells in need. In plants, vital substances like water and minerals are provided by the soil and are absorbed via the root, while the glucose is produced in the leaf during photosynthesis. The vascular tissue allows the substances to move from certain parts of the plant to the others. The two main vascular tissues in plants are the xylem and the phloem. If you have ever seen a fallen tree and take a closer look inside, almost all of its radical content and consisting of hardwood and sapwood are actually xylem tissues, and the inner part of its bark is the phloem. Now xylem. Xylem is responsible for distributing water and minerals taken by the roots. An interesting fact about xylem is that some parts of it do not have protoplasm or cell walls, allowing water and minerals to pass easily. Xylem consists of tracheids or conducting cell, vessels, fiber, and parenchyma. The water in tracheid does not flow continuously. The roots and leaves are connected through a series of tracheid cells with pitted walls. In contrast, the xylem vessels are long, wider, hollow tubes continuously stretching from the roots to the leaves. The fiber cells function mainly as supporting structure, and the parenchyma, the only living cell of the xylem, help with the food storage. The xylem cells are strengthened by a substance called lignin. The pattern of lignin will vary depending on the location. It can be a ring, pattern, spiral, reticulate, or pitted. Phloem. The phloem transported manufactured food like sucrose and amino acids from the green part of the plant like leaves to other parts of the plant. The process is called translocation. Phloem consists of sieve tubes, companion cells, fibers, and parenchyma. A sieve tube consists of a single row of elongated thin-walled cell. Just like the name suggests, it has walls perforated by nude paw like a sieve. A mature sieve cell has a thin layer of cytoplasm which conducts manufactured foods from one cell to another. The sieve tube cell has a degenerated protoplasm, which means that it needs another to carry out its vital processes. This is why a sieve tube cell has a companion cell. The companion cell has everything that the sieve tube doesn't, such as abundant cytoplasm and nucleus. It has an extremely thin but flexible cell wall, so that there is no hassle in keep the sieve tube cell alive. Just like in xylem, the fiber cell structural support and the parenchyma stores the food and other substances in phloem. The fiber cells are the only type of cell that is dead. Arrangement of vascular tissue. The vascular tissue has different arrangement in various parts of the plants in the roots of the herbivorous dicot plants. The xylem forms like an X shape in the middle, while phloems fill around the xylem. The xylem and phloem in the stem are clustered into circular shape. These clusters are located near the edge of the stem, while in the leaves, xylem and the phloem are located in the vascular bundle with the xylem positioned above the phloem.
Next is means of transport in plants. Plants rely on three primary methods of transport: diffusion, facilitated diffusion, and active transport. Diffusion is a passive process which involves the movement of molecules from areas of higher concentration to lower concentration, occurring within cells, between cells, or over short distances such as from the spaces within a leaf to the external environment. While typically observed in gases and liquids, diffusion can also happen in solids, although less frequently. Factors such as concentration gradient, membrane permeability, temperature, and pressure influence the rate of diffusion. Facilitated diffusion utilizes specialized proteins to help molecules cross membranes without energy. Types include symport, antiport, and uniport. Now talking about these three types, first is symport. Both molecules cross the membrane concurrently, proceeding in the same direction. Second is antiport. Molecules cross the membrane by moving in opposite directions to each other. Third is uniport. The passage of a molecule across the membrane occurs independently, not influenced by other molecules. Active transport requires energy, ATP, to move molecules against their concentration gradient, often facilitated by membrane proteins called pumps. Pumps are a type of protein that uses energy to transport substances across the cell membrane, including from low to high concentrations, uphill transport. The transport rate is maximum when all protein transporters are in use or saturated. Carrier proteins, like enzymes, exhibit specificity in the molecules they transport across the membrane and are sensitive to inhibition by compounds interacting with their side chains. Next topic is what is water potential. Water potential is the potential energy of water in a system compared to pure water, when both temperature and pressure are kept the same. It consists of two main factors, solute potential, Psi-S, and pressure potential, Psi-P. Now let's understand the work of water potential in detail.
So right here I have a container of water that is open to the atmosphere. It's standard atmospheric pressure up here, and let's just assume that everything in our system, the air and the water and the container, everything is 21 degrees Celsius. Now our chamber is, or our container of water is divided into two chambers, a left chamber and a right chamber, which is divided by semi-permeable membrane. It's permeable only to the water but not to the solute in the water, and on the left side we have pure water, so it's just molecules of H2O. You could view that a distilled water, while on the right side we don't have pure water. We have some sodium chloride that has been dissolved in the water, and we want to get a little quantitative in this video, so let's say it is 0.25 moles per liter or 0.25 molar sodium chloride. Right over here we know intuitively from a study of osmosis what is going to happen here. Since our membrane is only permeable to water and the right side is hypertonic relative to the left, it has a higher concentration of solute. We will have the water going from left to right, but what we are going to do in this video is get a little bit more quantitative and thinking about, well, what is the water potential of water to go from the left to right. And to do that we are going to talk about something known as water potential, which is denoted by the Greek letter psi. Now by convention, the water potential of the pure distilled water when it is in an open container exposed to standard atmospheric pressure, that is going to be, so psi over here is going to be equal to 0 bars, and as you can see here the units for psi for water potential are the same as the unit for pressure. Here we are using bar, but you could use psi or atmospheres or something else. But what is going to be the water potential on the right hand side? Well, generally speaking, you can break down your water potential into subcomponents, and the subcomponents that we typically talk about it, but there are more, are the component due to the solute and the component due to the pressure. This will often be known as the solute potential and the pressure potential. Now because this right chamber is open to the atmosphere and its standard atmospheric pressure, the pressure potential in this situation is going to be equal to 0. So our total water potential on the right hand side is going to the same thing as our solute potential. Now to figure out our solute potential, we will turn to a formula, and I will show you a formula sheet right over here that's given by the college board when folks take the EP biology exam. But we will see that this formula is actually quite intuitive and related to the ideal gas law that you first studied in chemistry. It tells us that
our solute potential is going to be equal to I, and I will talk about what that is in a second, times per molar concentration, C times our pressure constant R. Now they are calling it the pressure constant in this concept, but it's actually the same R that we will see in the ideal gas law times the pressure in Kelvin. So times T. So first let's start with I. So let me write this down. This is going to be equal to, it's all going to be negative because of these are going to be positive values. So I is known as the ionization constant, and one way to think about it is for every molecule of solute, how many molecules does it dissociate into when you put it into the water. So if I take a molecule of sodium chloride, we know that when we put it into water, it's going to dissociate into sodium ions and chloride ions onto 2 ions for every 1 molecules of sodium chloride. So here our ionization constant is going to be equal to 2. Now what is our molar concentration? Well, we already know that our molar concentration is going to be 0.25 molar, or we could write it as 0.25 moles per liter, and then we are going to multiply that times our pressure constant, which is the same as the ideal gas constant, and that is going to be, they gave us to write over 0.0831 liter bars per mole Kelvin, and then when we are going to multiply that times the temperature in Kelvin. Now what's that going to be? What's going to be our temperature in Celsius plus 273? So you had 273.21 plus you are going to get 294 Kelvin, and so this is going to be approximately equal to, we are going to have this negative out front. And if we look at our units, let's see, we have moles cancelling with moles, liter cancelling with liters, and then Kelvin cancelling with Kelvin. So units are going to be in bars, which makes sense because that's, we already talked about, that's the unit that we are dealing with here. And so let me get a calculator out. See, we have already the negative 2 times 0.25 times 0.0831 times 294 is equal to, and I will just round to one decimal place, so 12.2, but I have that negative there, so approximately negative 12.2 bars. So our solid potential over here is minus 12.2 bars, or it is approximately equal to that, and that is going to be equal to our total water potential because the pressure potential is 0. So how do we interpret that? Well, one way to think about is that you can side, that is, it will pull water in that the water, if the membrane is only preamble to the water, is going to from high potential to low potential, and that's exactly what we thought what would happen intuitively now. Another thing that we could think about is, well, how would we keep the water from flowing? Well, then we could add some pressure potential right over here. Maybe we could put a piston over here, and we could apply some pressure over beyond the standard atmospheric pressure. Well, how much pressure would we have to apply here to kind of push down on that water so that we don't have a net inflow from the left to right? Well, imagine if this was 12.21 to 0.2 bars that we were to apply here. Well, then all of the sudden this pressure potential wouldn't be 0, it will be plus 12.2, and then on the right side we would have minus 12.2 bars of solid potential plus 12.2 bars of pressure potential, and so that would add up to be a 0 water potential, in which case, since the left side and the right hand side would have the same water potential, would not have the flow of water in either direction. So as you can see, this idea of water potential allows us to think a little bit more precisely and a little bit more quantitatively about these idea of osmosis and tenacity.
What is osmosis? Osmosis is the movement of water across a selectively permeable membrane from areas of higher water potential to lower potential. This movement is influenced by pressure and concentration gradients. Isotonic solution, osmotic pressure inside and outside the cell is equal. Hypotonic solution, the external solution has a lower solute concentration than the cytoplasm, causing water to move into the cell. Hypertonic solution, the external solution has a higher solute concentration, causing water to leave the cell. We have studied little bit about osmosis in water potential. Now understand osmosis more in detail.
Water moves into and out of cell by osmosis. Picture an animal cell in solution. Let's take a close-up look near the cell membrane. There is plenty of water inside and outside of the cell, but there are also other materials dissolved in the water too, like ions, sugars, etc. Cell membranes allow water to pass through relatively easily, but they prevent some other materials from passing through so freely. A membrane that allows some things to move through but not others is known as partially permeable. We will just call these other materials solutes from here on in, and you can see in this diagram that there is a larger proportion of solutes inside the cell than outside it. We could say the solution outside the cell has a lower solute concentration, and inside the cell it has a higher concentration. This means that water is present in a higher proportion outside of the cell compared with inside of the cell. Where water is present in a larger proportion, we can say that the solution has a higher water potential. Obviously, we could say that the cell has a lower water potential in this diagram. What I will move from an area of higher water potential, the solution with a lower solute concentration, to an area of lower water potential, as long as there is a partially permeable membrane to prevent the solutes from also moving. The definition for osmosis is therefore the net movement of water from an area of higher water potential to an area of lower water potential through a partially permeable membrane. Note that it is also correct to define it as the net movement of water from an area of lower solute concentration to an area of higher solute concentration through a partially permeable membrane. Both definitions express the same thing. Let's see what happens when we place animal cell in solution of different concentration. If the solution has equal water potential to the inside of the cell, there will be no net movement of water and no observable change. If placed in a solution with a higher solute concentration, water would exit the cell by osmosis. The cell would shrink and become shriveled. If the cell were placed in a solution with a low solute concentration, water would enter the cell. And if the concentration of the solution with low enough, then enough water would fill the cell until it bursts open. The water movement is the same with plant cell, but the results are slightly different. In a solution with a high solute concentration, the cell can begin to shrink as water exists. If the solute concentration is high enough, the water content of the cell reduces enough until the cell membrane pulls away from the cell wall. This is known as the plasmolysis. Here is an image of onion tissue under a microscope. This onion tissue has been placed in a solution with a very low water potential, so water has exited the cell. If we look closely at a single cell, we can identify its cell wall, and we can see that the cytoplasm has shrunk due to water loss, and the cell membrane has pulled inwards from the cell. The cell has become plasmalized. In lower solute concentration, plant cells will increase in size as they take in water, but even in very low solute concentration, plant cells won't burst. The way animal cells do this is because the animal cell wall which surrounds the membrane is really strong and it won't split under the pressure of the water. Make sure it's clear in your mind that animal and plant cell have a cell membrane, but it's cell wall of plant cell that prevents them bursting. That animal cell don't have a cell wall. When discussing osmosis, it is sometimes useful to use the words hypotonic and hypertonic. Hypotonic can describe a solution with a water potential that is higher than it is inside the cell. A hypertonic solution is just the opposite. And by the way, there is a word we use when the solution has an equal water potential to the cell. We would say the solution and the cell are isotonic. If you struggle to remember which picture a cell being dropped into a hypotonic solution become more O-shaped as it takes in water. If you want to observe osmosis, it's obviously not very convenient to place a single tiny cell in solutions and see what happens. But what we can do is place larger tissues in solution. If we take strips of potato, which are made up of plant cell of course, and place them in different solution, we would notice a physical change after 20 minutes or so. The strip placed in a solution with a high solute concentration would lose water and become soft or floppy. We would use the word flaccid to describe them. If placed in a low solute concentration, water would enter the cell of the tissue by osmosis, making it swell up and become firm. Here we would use the word fugate to describe the firm texture of the
Plasmolysis. Plasmolysis is the process where plant cells lose water in a hypertonic solution, causing the cell membrane to pull away from the cell wall. It is reversible if the cell is placed in a hypotonic solution, where water re-enters, increasing turgor pressure and restoring cell rigidity. Next topic is imbibition. Imbibition is a type of diffusion where solids, such as seeds, absorb water, causing them to swell. This process requires a water potential gradient and an affinity between the absorbing material and water. Pathways of water absorption in plants. Water absorbed by root hairs follows two pathways to reach the deeper root layers. Apoplast pathway, water moves through cell walls and intercellular spaces. Symplast pathway, water moves through the cytoplasm of cells via plasmodesmata. Transpiration. Transpiration is the loss of water through evaporation, mainly through stomata in leaves. It helps with nutrient transport, cooling, and water uptake. Factors affecting transpiration include temperature, light, humidity, wind, stomata number, and stomatal opening. Understand transpiration in detail.
Transpiration. We just studied that the transportation of food in plants occur in phloem and its water occur in xylem. Now we will discuss transpiration and the fact it's affecting it. Plants continuously absorb water from the soil, but not all the water absorbed is used by the plant. Excess water evaporates from aerial parts of the plant and mostly through the stomata below the laminar of the leaves. This process is called transpiration. Ever wondered how plants draw water against the force of gravity? The evaporation of water from leaves results in a suction force which pulls water up the xylem vessels. The suction force is called transpiration pull. It is the main force that pulls water and mineral salts up the plants. This upward movement of substance is called a transpiration stream. Factors affecting the rate of transpiration. Transportation depends on evaporation. Therefore, factors affecting the rate of evaporation also affect the rate of transpiration. These factors affecting the rate of transportation are humidity, temperature, strong wind, and light humidity of air. The intercellular spaces in the leaf are normally saturated with water vapor. If the outside air is dry, water vapor will diffuse more rapidly out of these leaves. That is, the rate of transpiration will increase. Whereas if the air is damp or humid, evaporation is limited. The more humid the air is, the slower is the rate of transpiration. Temperature of the air, assuming all other factors remain constant. A rising temperature of the surroundings will increase the rate of evaporation, and thus transportation rate also increases. Strong wind when the air is still transpiration makes the air around the leaves moist. The water vapors build up and increase the humidity, hence, as we have started before, reducing the transportation rate. During a windy day, the moist air around the leaf is blown away, making the air less humid. This increases the rate of transportation. The stronger wind, the higher the rate of transportation. However, if the wind is very strong, the stomata may close because the guard cell lose too much water. What would happen if there is a cool, wet wind? Light affects the size of the stomata opening on the leaves, thus affecting the rate of transpiration. During the night or in the shade, the stomach are closed and reduce the rate of transpiration. But on a sunny day, the stomata open wide and increase the rate. The movement of water vapor outward also remove heat from the plant, thereby cooling the leaves and preventing them from being scorched by the hot sun.
Thanks for watching the video. We will drop the notes in the description.